All right. So today we got chapter 3, and this will kind of dovetail into the lab macromolecules, food molecules. It's the macromolecule lab. Excuse me, I have really bad allergies today. Some things that you should be able to kind of understand what goes into all of these organic molecules. And we're only talking about kind of, well, the 4 major ones. Really 3 is all, from a nutrition standpoint, from an eating standpoint, is really all you care about. But we're going to talk about the 4 major macro organic molecules that we have: carbs, fats, lipids, and protein. And then nucleic acids, the number 4. When you think of organic molecules, those are the 4. But when you think of organic molecules on the daily, really it's the macros. It's carbs, lipids, fats, and proteins. That's really all you think of. So there's a little bit of nutrition kind of sprinkled into this lab, or I'm sorry, to this lecture here today. So you should be able to discuss all of these guys, okay? Carbs, lipids, proteins, nucleic acids. I'm going to be going over what makes these up. Some of them are really basic. Some of them can be added to and get really complex in structure. Again, you should be able to do that. Give a specific example as well. Those are all things that you should be able to do from this lecture. So we're talking about organic stuff. So when we talk about organic stuff, it really contains one major thing, and that's the C word, carbon. It also has a bunch of hydrogen atoms on it, but water has hydrogen atoms on it, and it's technically not organic because it doesn't contain that guy right there, carbon. That's the big thing. All organic molecules share carbon, okay? It's a commonality between them all. Carbon is really easy to manipulate. It has a lot of open spaces, open bonds available to it, so it can react with a lot of different things. So that's what makes carbon so cool in this regard here. But this book refers to these organic molecules as biomolecules. Okay, sure. I mean, this is literally the only place you're going to see it. Okay, but they are the 4 classes of organic molecules, these quote unquote biomolecules: carbs, lipids, proteins, nucleic acids. Okay, functions are going to be highly varied. It will depend on the subtype of carbohydrate and what it does. Different lipids, various different examples of different lipids do various different things. Proteins can be really basic and simple, and they can fold and be really super complex and do a much greater number of functions in the body, varied functions in the body. Nucleic acids are pretty straightforward in what they do, and we'll get into all of these as we continue through the lecture today. Again, there's only 6 electrons, okay? 4 in that outer shell, that valence shell, okay? Remember valence, V-A-L-E-N-C-E, okay? That valence shell. So it has 4 open spots. So because it has 4 open spots, it can bond with a number of multiple things: nitrogen, hydrogen, oxygen, phosphorus, sulfur. It can do a bunch of different things. So we can manipulate this thing. It's this carbon-carbon bond, very stable. Carbon-carbon double bond, even more stable. Okay. You can connect a bunch of carbons together and form these really long hydrocarbon chains, and that's kind of the structure behind lipids and how you can make medium chain and short chain and long chain fatty acids. Okay, it's that ability to easily put a carbon in there. Okay, easily bond carbons together. Besides from double bonds, they can do triple bonds as well, and it's just represented like this. Okay, that's super strong. Not a lot of movement can happen. It does create a kink, and we'll see this concept when we start looking at lipids here in a little bit. Aside from making long hydrocarbon chains, they can form these very complex branches it could even form what's called a ring structure, and that's a basic ring structure right there. Carbon, 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 okay, at each of those points. And you see a lot of ring structures with hormones, testosterone, estrogens, very similar. They have these carbon ring structures associated with them, and then they have some branches that come off of the rings as well, and that's what gives it that unique characteristic to be estrogen or testosterone, depending on your body. All of these different things, nitrogen, hydrogen, oxygen, phosphorus, all of those things hang off that carbon chain. And those things that hang off that carbon chain are what give all of these structures that carbon can form their very specific marching orders, okay? Their specific function, their specific way they're going to react with different things. This is called a functional group, and it determines two things, okay? The big one is chemical reactivity. There's an entire just class basically in chemistry that looks at chemical reactions for organic molecules. It's mind-numbing, okay? And there are so many different types of chemical reactions with those specific functional groups. Understanding those, then you can understand how you can manipulate things in a lab setting to form different structures. So chemical reactivity, that is a huge characteristic that these functional groups impart to the carbon skeleton, okay? So that's going to help determine function, and that's going to help determine its structure, okay? All because of this little bitty functional group there hanging off. What does that look like? Here, okay? There's the functional group. There's the functional group. They're isomers. These have identical molecular formulas. They have the same number of carbons, the same number of hydrogens, the same number of oxygens. They are just in a different arrangement, okay? And by changing the arrangement, I change those functional groups as well. A glyceraldehyde to an acetone. And it gives it a completely different reactivity. Okay, this is super reactive over here on this side, this glyceraldehyde, super reactive. Okay, and again, it's just that little functional group. We're not going to get into kind of going over every single one of these little functional groups. We do a little bit maybe in nutrition class in terms of proteins, but we're not going to get into all that complexity here. We have a lot more things to scare you with. All right, so all of these biomolecules, these macromolecules, I call them macromolecules. Okay. They're biomolecules because they're organic, okay? So that's the term that the book uses. I call them macromolecules because these things are generally big, right? They are big things. Carbohydrates are big. They can be complex, right? Complex carbohydrates. A very long chain of glucose molecules. Lipids are multiple different things put together, okay? So that makes it big. Same thing for proteins. Generally, proteins are very long chains of amino acids. Nucleic acids are several different things congealed together as well. So because they are so big, I like using the word macromolecules, okay? Macro means big, okay? So these are big molecules, but they do a bunch of fantastic things. So we need them big because then we're going to break them down and take those component parts, and now we're going to use them inside of us to build cells, to make different things, to make some energy, some ATP. So these macromolecules usually consist of smaller and smaller things. Those smaller and smaller things are referred to as a monomer, okay? You're going to need to know what are the basic component parts, what are the subunits, what are the monomers for all of these macromolecules, okay? What are carbohydrates made of? Lipids made of? Proteins made of? Nucleic acids made of, okay? So you're going to need to know the monomer, okay? These macromolecules are polymers. Poly means many, okay? Many. Mono means one. Okay. So if I go over here to this slide here, carbohydrates, that is the basic subunit. That is the monomer, a monosaccharide. And there are a few different kinds, and we'll get into that when we start talking about carbs here in a second. Okay. The monomer for lipids are glycerol and fatty acids. Subunits, the monomers for proteins are amino acids. And just one little monomer of a nucleic acid is a nucleotide, but there's some basic other components as well that we need to form the polymer, DNA or RNA, okay? So these are the monomers. Okay, you will need to know that. You'll probably be asked on an exam. I know you'll probably be asked on that lab assignment as well. If we were to do this lab in the lab setting, face-to-face this semester, that would have been what we were testing for, okay? You would have been given an unknown, and by doing various different tests, you could have determined the content of that unknown and then told me it was, you know, milk or soda or something along those lines, okay? So these monomers are important to remember and understand because it is these monomers that give it the reactivity that we would have seen. Now monomers get strung together and we make a polymer, okay? This is basic building block, okay? Are these monomers. This is the complex structure. Okay, so polysaccharides, complex structure. Fat, complex structure. Okay, polymer. Many different glycerol and fatty acids go into the fat. Same thing for polypeptide. I'm just stringing a buttload of amino acids together. DNA and RNA, like I mentioned earlier, there's a handful of things that go into the structure that make it DNA and RNA. Nucleotides are the basic monomers behind that though, because we take those out, and now it's just nothing that can function. So those are the very complex structures that actually do a heavy amount of work within the body. To get all of these things together, I need to create a chemical reaction, okay? I have a dehydration reaction. Dehydration reactions take those subunits and brings them together, okay? Forming a covalent bond, okay? So they are sharing electrons here, okay? Remember, covalent share, ionic takes, okay? Water is produced, all right? There are different ways to kind of visualize this, okay? It's called a dehydration reaction because I'm removing water from the basic building blocks, okay? So this is usually called a dehydration reaction because I'm having water and it's being produced and I'm removing it there, okay, from those two basic monomers, from those two little molecules. So it's referred to as a dehydration. You can sometimes refer to this as condensation because water is produced in the process. It says it right there, okay? So some textbooks will refer to this as a condensation reaction because water is produced and is removed from the reaction area. So condensation is another name for this one. I'll write it over here for you. Condensation. Okay. Dehydration reaction, because water is removed from the reaction. Or some books refer to it as condensation, because water is produced and it does leave. So different ways to kind of visualize it. All right. This is used to build. Okay. A dehydration reaction is used to build. Okay. It connects monomers together. It makes something big. If I go over here, okay, here's, there we go. Okay. There's my monomer. There's my monomer. Okay. Right there and right here. So there's my two monomers, right? Water is produced and water leaves the reaction. And I've plucked those two together. These two go together, obviously. And that bond come together and ta-da, there's your new little biomolecule. Conversely, if I add water to those reactants, okay, that is called hydrolysis. Look at the word. In science, look at the word, break it down into its component parts, okay? If I look at this word, I have hydro, and that's water. And the other word is lyse, and that means to break or destroy. To break, destroy everything. So I add water. Water is added to the reaction here. It's added to those reactants, and I can break it up. And it breaks down polymers into the individual subunits. Okay, here's my here's my biomolecule right here. Okay, I add water to the situation, so I'm adding it in. Okay, and there it's just the exact opposite of a dehydration reaction, right? Okay, so dehydration reaction is making something bigger. It's making a macro. Okay, it's making a macromolecule. Hydrolysis, add water to the reactants, or to the reactant in this case, and it destroys it. Okay, it breaks it down into its component parts, into those monomers. Being able to maybe explain the difference, that would make a good potential short answer question. Sometimes it does, sometimes it doesn't. Which is to say that sometimes I have it as a short answer question and sometimes I don't. All right. There are other things that go into this chemical reaction process. And that other thing is enzymes. Enzymes, their big thing is that they speed up chemical reactions. That's what they do. Okay. Enzymes bring the reactants together faster. Okay. So if these two guys here are going to come together and react, enzymes just facilitate it a lot faster. So enzymatic reactions can go a lot faster. I can break down different things, or I can destroy different things, or I can make ATP and I can energy, okay, and I can make energy a hell of a lot faster when I have enzymes involved. And when we look at when we start to look at metabolism, we're going to see that concept, cellular respiration, where we start to look at that concept of enzymes and really what they can do for us and what they do for us in terms of bringing things together faster to make energy. That's the hallmark behind enzymes, or I'm sorry, vitamins. Vitamins make a lot of enzyme components. So vitamins, why are they so helpful and beneficial for making energy? Because they themselves don't provide energy at all, but they create the components for different enzymes. And those enzymes help facilitate and speed up enzymatic reactions, like when we need energy. That's why 5-hour energy is so fantastic, because it's filled to the brim with B vitamins. And B vitamins help make enzymes. A lot of them. Not consumed in the reaction. They're not changed by the reactions. They just are catalyst, okay? So they're never used up. They're never destroyed. Their shape is never changed at all, okay? They bring the two parties together. Reaction happens. They go out and find two more people to bring together, okay? Catalyst, speed it up. RxNS is shorthand for reactions. All right, and we'll be looking at enzymes a little bit later as well. So we got our first little macromolecule, biomolecule, whatever you want to say it, however you want to say it. Carbohydrates, their major function is to make energy. Okay. Carbs, number one goal. Carbs do a number of different things. Okay. They can help make building material, which, it goes into kind of the shell, contains chitin. And here's what that kind of chitin looks like here on the cell wall of a bacteria. Here's a shell, contains chitin. Okay. Cell wall contains cellulose, which is a carbohydrate derivative. Okay. So this hard exoskeleton that you see on a crab, carbohydrate, is the basic structural component behind that sucker. So it does have a structural role. It makes different things for various different organisms, as you can kind of see there. But for us, why do we want to get carbohydrates in our diet? Because it helps to make energy. And that's what energy is. ATP, adenosine triphosphate. I think we have that at the very end of the lecture. Okay. A 1 to 2 to 1 ratio. I'm not gonna ask you that. But there is a ratio. But just pretend you didn't see it. We have a lot of different varieties of carbohydrates. Okay. We have monosaccharides. Mono means one. Saccharide means sugar. We have disaccharides. Two sugars strung together. Okay. And then we have polysaccharides. Many sugars. Okay. So again, look at the word. If you can't figure it out, break it down into its component parts. Okay. Mono means one, di means two, poly means many. Okay. If it was a trisaccharide, that would be three sugars strung together. Okay. But anything above two is generally just referred to as a polysaccharide. So these are our three varieties. These, yes, you should know. All right. So when someone says, I'm going to consume a simple sugar, what the hell is that? Well, it's a monosaccharide. That's what they're talking about, okay? Simple sugars are monosaccharides. Monosaccharides and disaccharides are technically kind of clumped together and considered simple sugars, okay? We have three different ones, okay, that are the major ones that people think of when they think of a monosaccharide. Glucose, fructose, and galactose. All of these are very similar. Okay, that's what it looks like. That's that ring structure. Okay, C6H12O6. This is glucose. Okay, they all have a very kind of similar structure. The 6 carbon atoms. Okay, glucose is your blood sugar. This is from a dietary standpoint, this is one of the bigger ones, okay? Whenever you eat complex carbohydrates, it is really a multitude of a lot of different glucose molecules strung together. So that's what your body's primarily utilizing. That's the primary sugar found in the blood as it's being transported around. Fructose is fruit sugar. So if you consume fruits, this is what you are getting a lot of. And high fruit consumption is not a bad thing. Okay. In fact, I think like something stupid like 2 or 4% of people in the United States really get the requisite amount of fruits servings in a day anyway. So if you're worried because you may get the diabetes, that ain't gonna happen just by consuming fruit. Okay, fruit's not that bad for you guys. Galactose is another one. Chickpeas is kind of where you'll find that. We have a couple 5-carbon sugars. Okay, these are 6. We have a couple 5-carbon sugars. This is where we're going to talk about in nucleic acid. So we'll talk about DNA and RNA at the very end here. And that's where you'll see those monosaccharides, right? But really, these are kind of the big ones here. When you think of monosaccharides, these are the big 3 that you think about, because they're primarily the ones in your diet that you're consuming. You're not really going to say, oh, how much deoxyribose is in my meal. You don't do that crap. Okay. Glucose, fructose, galactose, those are the primary monosaccharides that you think of because they're in our diets. So we have those little dehydration reactions that we talked about, and that's how we get the disaccharides. Now, that one you probably know, and you probably know it as that. Sucrose, you know it as your table sugar, okay? What is lactose made from? Because lactose is a disaccharide, so I can break it down into its component parts. I can break those monomers up, and I get a molecule of galactose, and I get a molecule of glucose. It takes a specific enzyme, right? Lactase. So if you're lactose intolerant, you don't produce enough of the enzyme to break down that milk sugar. And just causes, upset stomach, bloating, gas, all that fun stuff. Sucrose is your table sugar. It is composed of a molecule of glucose and a molecule of fructose. And in the lab for this biomolecules, the food molecules, you would be able to determine if you had a sample of table sugar or a sample of milk, okay? And you would be able to determine if it was sucrose or if it was lactose by figuring out what's in it. And it's just a different test to look for these specific monomers. So it's pretty fun. Maltose is 2 glucose molecules, okay? So maltose adds a hell of a lot of glucose into the system, right? Maltose you'll see in different beverages and whatnot, and 2 glucose molecules. So, if you're consuming a lot of beverages with malt in it, you are bombarding that system with twice the amount of glucose molecules. That's why if you drink a lot, that's why you get that little tire around your midsection, because of all that sugar, and it gets converted to the F word, and no one likes the F word. The F word is fat. The last of our carbohydrates here is the polysaccharides. And poly means many. I love to ask questions where they are one-word difference, okay? In any board exam, to be in any field of medicine, any board exam to get certified to do literally anything in life, to get a license to practice that profession, all of the questions are, a vast majority of those questions, I guess I should say, ask you something where there is one word difference between them. So I like to ask those kind of questions because I've taken a billion board exams, I feel like. Our first 2 examples of polysaccharides, many glucose molecules jammed together, and that's what you're seeing right here, okay? very branched, not branched, very complex in structure. Okay. Starch is how plants store energy. Okay. They get that by, when we'll look at photosynthesis, and we'll see kind of how they utilize carbohydrates to make different things. Okay. Or utilize it to do fund activities inside of those cells. So we'll look at that when we look at photosynthesis. But starch is the energy storage form in plants. Inside of us is glycogen. Okay. When you consume glucose, all right, and it's flooded in your blood and it's transported around to cells, okay, it will take that glucose to the cell and be like, here, I have the component parts for you to make energy inside you. So those cells will take it up. When all the cells are filled with glucose, then they don't come in the cell. So the body changes it and converts it into glycogen. And it's the storage form of sugar in the body, basically, okay, for us. So when you're, let's say about, I don't know, a couple hours after you eat and your sugar levels start to diminish, okay, because it's being converted into glycogen and put into storage, okay, and now the cells are like, you know, an hour, 2 hours later, they're like, man, my tank's getting empty. I need some carbohydrates. I need some glucose to make energy so I can still do stuff. The body releases a hormone, pulls it out of storage, sugar levels rise back up in the blood, and now it can be distributed to the cells. Okay? And that's what really the pancreas does with insulin and glucagon, okay? Helping to transport and get that in and out of storage. So that's what that pancreas is doing there. Those are kind of clumped together. Those two are pretty important. The other ones are more structural related, okay? Energy. These are structures, okay? We have cellulose found in the cell walls of plants. Chitin found in the cell walls of some fungus, some fungi. And that little picture that we showed with the crab, it's also the exoskeleton of a handful of animals. Peptidoglycan, cell wall of bacteria. Okay, so these guys here are more structure. Okay, and these guys are energy. All right, so these polysaccharides are highly varied in what they do based on the structure of these guys. Okay, you have two forms of polysaccharides that are involved with energy storage, and then we have three of them involved with structure, making things. Peptidoglycan, cell wall in a bacteria, which, you know, helps to protect it. Okay, gives it its unique identifying characteristics, so we can, you know, treat it with specific, you know, medications, antibacterials. Cellulose, structure, cell wall in a plant, gives it rigidity, gives it structure. Okay, be the hallmark for, you know, why a plant can stand up the way it does. Chitin, found in the cell walls of fungus, found in the exoskeleton of those animals that produce that structure. Okay, so structure and energy. Okay. And there's what it looks like. Okay, that's what a cellulose fiber looks like. That's what the roughage looks like. Okay. So hydrogen bonds, as you can kind of see, they're holding these things together. So those bonds that we talked about last week, well, you know, we're seeing them here, okay, helping to make this structure. Lipids, there are a multitude of various different structures associated with lipids. So they're kind of all over the place. They are very large and they are insoluble in water, okay? So they don't dissolve in it. Because they have such a varied structure, they have a multitude of various different functions. Lipid is fat, okay? So that's probably what you're thinking. And, you know, Long-term energy storage and protection kind of go hand in hand with the F word. Long-term storage, anything your body is not using, the body converts it to fat, and then it keeps it there for, that rainy day. If you're not consuming food, then it pulls it out of your fat stores and provides sustenance for cellular activity, which we'll look at a little bit when we talk about metabolism later. It's also a concept we go over a hell of a lot in nutrition as well. It is cushioning, so it is going to provide some protection. A little bit of fat around organs is not a bad thing. A lot of fat around organs is a disastrous thing. If it's excessive, okay, in that fat around the organs, then it changes their metabolic activity. And that leads to metabolic syndrome and a whole bunch of nasty things that you really don't want to have in life. It does provide some heat retention. It's an insulator, okay? It insulates. Okay, it insulates. So it is going to keep you warm. It does have some structural components to it, and we'll see that with the cell membrane here. It's going to help define what a cell is and the components within that structural component, even add the fat inside the fat structure, even add some more functionality to it. So a lot of cool things there. And then it's involved with communication and regulation. Okay. So it wraps around nerves, and it helps to speed up nerve impulse conduction, okay? How information gets from your brain, to your hand, right? So it speeds that up. How much? If there's no fat around nerves, nerve impulse travels at around, I don't know, a couple miles per hour, which sounds fast. But if you put fat around a nerve, then it goes into that impulse can travel at around 200 miles per hour, or roughly around there, depending on the size of the nerve itself. So this concept of fat helping with cell communication and regulation can really help speed up how the body communicates with itself. So a lot of different varieties there. Fats, oils, phospholipids, steroids, waxes. So, a lot of different structure. We said it was a varied structure. With a varied structure comes the varied and the function component there of it. So here's that part that was part of that plasma membrane. It defines what a cell is. And I believe that's the next lecture. Yeah, we talk about the cell in the next lecture there. And we'll talk about one of the components being the plasma membrane. I think that's actually chapter 5. It's its own individual chapter. Phospholipids, so it's a component of plasma membrane, so it's going to help define a cell, transport stuff around within the body. Steroids do a lot of fantastic things. They make your sex hormones, so they make testosterone, they make estrogen, they make cholesterol, and cholesterol is needed. Cholesterol is not that bad of a thing. You need some. If you consume nothing but cholesterol, then you're in a world of hell. But cholesterol by itself is not necessarily a bad thing because it helps to make some of the components of estrogens and testosterones. Waxes, they're kind of weird in their regard. They have a weird shape. They usually prevent water loss, so it's involved with protection. Fats and oils there, as you can kind of see, one's a long-term energy storage in us, fat. Oils do it in plants. Okay. When we look at our first example of a lipid, triglyceride, okay, when you think of food, and I bring everything back to food, when you think of fats, triglyceride is what you're thinking of, okay? This is kind of what you're consuming in your daily diet, all right? Your fats, your oils, triglycerides is what you're getting in, is what you're really calculating ultimately, all right? And it is, I'm looking more of it from an animal, okay, because we're animals, right? So I look at it more from the human side than the plant side. So if you're a plant person, I apologize. I like plants. I just like the human body a lot better. When you look at triglycerides and you think of fats, you think of insulation, and you think of that long-term energy storage, okay? You know, different training methods allow you to kind of access these long-term energy stores, and you can diminish your fat content by exercising a certain different way. Kind of nutrition class, we talk about it there. But the big thing about triglycerides, okay, and these fatty acids, because remember it consists of one glycerol molecule and 3 fatty acid. Should probably write that there. 3 fatty acid chains. There it is. Okay. There's your glycerol. And there's your fatty acid chains. 1, 2, 3. Okay. Same thing down here. When it comes to triglycerides, they can be, as we just saw, saturated or unsaturated. Okay. Now you've definitely heard of saturated fats. Saturated fats are bad for you in huge amounts. So are trans fats. Okay. Saturated fats, why are they bad? Why are they bad? No double bonds. Okay. So they are saturated with hydrogen ions. So if I look here, here's my carbon backbone, okay, my hydrocarbon chain. And every possible bond with carbon is filled with a hydrogen molecule. It is this orientation here of saturating this thing with hydrogen bonds that gives it this very rigid structure. Okay? And so these have a tendency to be very solid at room temperature. Okay? And these are tend to be more bad for you in nature. Okay? Unsaturated will house these things called carbon-carbon double bonds. It can be 1, it can be 2, there can be many of them. Okay? These tend to be liquid at room temperature because of the presence of those double bonds. Those double bonds take away a hydrogen atom. Okay? So here, as you can kind of see up here, I am saturating this carbon with hydrogens. If I look down here, it's not saturated anymore. I have a carbon-carbon double bond there, so that takes away a bond for hydrogen atom to that carbon atom there. So now it's unsaturated. Okay. Carbon-carbon double bond. And when you put those carbon-carbon double bonds in here, you create these kinks. Okay? You create kinks in the chain by having those double bonds there. Because you get rid of hydrogen atoms, these have a tendency to be more liquid at room temperature. They have a lot more fluidity to them. They're open, okay? This is not open. I can't get into the inside here and do anything, but good God Almighty, look at this thing. It's wide open, okay? So there's a lot more fluidity to this thing, okay? Because it has those kinks in it. And so that's why these things are more liquid at room temperature. Sometimes these can naturally occur, these trans fat. And a trans fat is right here, okay? That's a trans fat. CLA, conjugated lipoic acid, CLA. It is a trans fat that naturally occurs. You have a naturally occurring trans fat in red meat. Okay. It's also something, trans fat is also something that we can make. We can Frankenstein a cis. Okay. And cis just means they're on the same side. Okay. Cis, same. Trans, opposite. Opposite. Okay. We can also make these trans fats, and that allows food to to sit on a shelf for 3 to 6 months. Okay. Which ensures that we never go hungry, which unfortunately people still do. Okay. One bond in the trans configuration. Trans opposite. Okay. Trans opposite. And here's, again, looking at the monomers, glycerol, 3 fatty acid chains, dehydration reactions sticking together. I probably should have put that earlier in the presentation. So that was triglycerides. Triglycerides is what you are consuming when you're eating your food, all right? Another major form of lipid are phospholipids. Very similar in structure, okay? Very similar in structure, okay? There's a hydrocarbon chain. It can be kinked. It doesn't have to be kinked. So glycerol. So pretty similar, okay? Got your glycerol molecule, but now we have only 2 fatty acids instead of 3, okay? So similar, but different, right? And we have a phosphate attached to it. And that's what you see right there. Phosphate, glycerol, and your 2 fatty acid chains. As you can kind of see here, okay, phospholipids will form barriers, form boundaries. And they have, in these 2 components, sorry, I just keep going fast, okay? This part here is the polar head, and this is going to be called the nonpolar tails. Because they have this nonpolar and polar component, they always like to, I don't know why that keeps jumping like that. They always form these barriers, and they always orientate with this head towards the water. Towards the water. Okay. If we break this word up, right? Hydro means water. And philic means love. Okay. So these phosphate heads are always going to be orientated towards the water. Okay. Here's the inside of the cell, filled with cytoplasm. Water, a component of it, right? Outside, next to extracellular fluid, a component of it is water. Okay. So these phosphate heads are always going to be pointed to the water. These phosphate heads are always going to be pointed to the water. And the lipid tails, okay, are always pointed away from the water. Why do they do that? Because they don't like water. Okay. Hydro, water, phobic, fear. All right. So they always form this double layered membrane because these guys don't like the water. They don't like to be exposed to it. Okay. Because if they are, it will destroy the structure of the cell membrane. We'll talk about that when we look at chapter 5. A little bit more in depth there. All right. So they always form these double layered membranes. Okay. This helps to form and define a cell. A cell is a basic structural functional unit of anything. Okay. So these plasma membranes help to form these cells. And that's what's going to give us tissues and organ systems in you. Okay. So they always form this double layer, okay, where the heads are always towards the water and the fats are always away from the water. Yeah. And you can see there's kinks, and we'll look at this a lot more in depth in chapter 5, okay? But it does give the cell membrane some pliability, allows these double bonds with these kinks allow us to handle varied temperatures a little bit better. So cellular structure doesn't kind of break down. So these kinks are a good thing, okay? These kinks are a good thing. They're a very good thing in terms of your nutritional profile, at least, as well. We have steroids and waxes are the last couple of categories that we have here, excuse me, when it comes to lipids. So triglycerides, phospholipids, steroids, and waxes, okay? Steroids are different and unique because they are the only one that have this ring structure, okay? There's a ring structure. There's the ring structure, okay? So this is cholesterol and then the sex hormones. And as you can see, we need cholesterol to make sex hormones. So we need cholesterol in our diet to be able to manipulate it and change it and form testosterone or form estrogen. Okay. The only lipid that has the carbon rings. Multiple different functions. We will find some steroids within the cell membrane. They're going to be involved with a multitude of regulation, and we'll talk about that later. But really, cholesterol, testosterone, estrogen are examples of steroids, okay? And cholesterol helps to make testosterone and estrogen. That makes a guy, that makes a girl. There you go. If you consume too much cholesterol, it'll accumulate within your blood vessels, and then that's going to what causes that that blood vessel becomes stiff and rigid. That gets you the blockage. That gets you your heart attack or angina, and that gets you dead. You don't want that. The last thing we have here for lipids is waxes. Waterproof. If you can think of beeswax, then you can probably visualize it that it's solid at room temperature. But you know, for the big one here for us guys, it's waterproof. So that's going to be protection. Okay, it resists degradation, it resists breakdown, it protects structure. Okay, we have a waterproofing agent on our skin. Okay, we have a couple different waterproofing agents. We have keratin that gets produced that gives us this waterproofing protection for our skin. We also have sebaceous glands, which produce a waxy sebum material, which keeps the skin always soft and supple so it can function properly. All right. So it provides some waterproof. It provides some protection. Okay. Where do we see it? You know, beeswax, okay, plant cuticle, okay, earwax in us is an example where you see a whole lot of it. Again, it's an opening to the outside world, so we have a waxy production there, cerumen, and that helps to trap anything that potentially tries to get in that way. It also lubricates the tube as well, the ear canal, allows sound waves to travel through a little bit faster as well. So there are some slightly other functions that we have here, but waterproof and functioning in protection. Okay. Helps to maintain that plasma membrane. Okay, which helps the integrity of the cell. Okay. And that helps to keep the structure. functioning properly. Okay, because if we don't have that waterproofing type of thing there, then you can absorb things through your skin that you may not necessarily want to. And we have some regions of the skin where we don't have a waterproofing agent. It's just on our skin. Okay, but again, I mean, you don't eat through. Well, I mean, I hope you don't. You don't eat via your skin. The cuticle, that skin on the fruit. And I imagine you can figure that out. That's your beeswax, right? All right. Our third macro or third biomolecule here are proteins. Proteins are very complex things. They are made from the subunit amino acid, okay, just by sticking them together. We stick amino acids together by a specific type of covalent bond, okay, and that's a peptide bond. A peptide bond is just a subset of a covalent bond specifically between amino acids. Okay. There are lots of them, as you can see from the slide there. We have a multitude of ways that we can string these together. And because of that, we have a multitude of various different functions and also a multitude of various different types of shapes. Okay, we have different shapes which give us the essential function for these things. What are proteins? What are their functions? These are their functions. 1, 2, 3, 4, 5, 6. It is these 6 functions that give us such a wide variety of different ways that we can connect amino acids together and get very different functions. The first one, metabolism, getting energy for us, okay? This is how we get energy, get ATP, okay? All right, that's how we get ATP. So they help to facilitate chemical reactions. We just talked about those earlier, right? Enzymes, okay? They're proteins, basically. Another major function is they make stuff. They make structural components. All right, so they make keratin. Okay, keratin is in your hair, it's in your fingernails. Right? It makes collagen fibers. Collagen fibers help to make connective tissues. Right? Collagen fibers help to make your skin look good. Okay? Transport. Multitude of various different specific proteins that carry specific things. Okay? Hemoglobin is an example. Okay? It's a very complex protein structure and it transports oxygen and carbon dioxide around the blood. We also have what's called membrane channels, and these are specific little channels within the plasma membrane of a cell that brings things across. Okay? Brings stuff from the outside, gets it into the inside. Defense. It makes antibodies. It makes a number of other cells as well, but antibodies is really good enough for you right now. Okay? This helps to keep foreign invaders at bay. Regulates. There are a number of hormones that are just regulatory proteins that say metabolism can go now, stop, go now, stop. So there's a lot of regulation involved here. Motion. Okay, obviously I can't do this and scratch my head if I don't have any protein fibers in all of these skeletal muscles that interact with each other, allowing muscles to move. And that's what actin and myosin is. Okay. Not only skeletal muscle, but the heart to pump, for food to move through your digestive tract, okay, because you don't control that, but it's the interaction of actin and myosin that cause that contraction, that cause that motion, that movement within the body. So here are my amino group, acidic group, R group, hydrogen. There are 20 different amino acids, and don't worry, I'm not going to ask you to know them. Okay. But it's that R group, okay, that gives it that, that gives it that very complex functionality to these various different proteins when we string them together. And here's what that looks like. These are all of your proteins right here. Some of these are essential, some of these are non-essential. And what the hell does that mean? It just means that you have to get them in your diet. Okay, your body can make some of these, it can't make some of them, and those are the essential ones. If you don't get them in your diet, you know, you don't grow. Okay? Failure to thrive, when a child is neglected and they're not fed properly. It's called failure to thrive, and that's because they are lacking these proteins in their diet. Again, just showing you all these dehydration type of reactions here. Proteins can be really complex, but you can destroy proteins, okay? You can change its shape and totally get rid of the function of that specific protein. If you cook hemoglobin, okay, if you heat it up, that destroys its shape, and now it doesn't bind to oxygen, or it won't bind to carbon dioxide. So hemoglobin will diminish in its functionality. And you get various different symptoms because of that, right? That's why our body has this complex buffer system to keep everything at homeostasis, and it functions in this. If it becomes too acidic, you destroy the protein structure. Okay. If it gets too alkaline, if it gets too basic, you alter the shape of that protein. So it is very important that we maintain pH levels, temperature levels, okay, within the body, so these proteins don't stop functioning. But this is how we cook, right? You cook with high heat. You break down the protein, that makes it edible now, and you can eat that hamburger and get something out of it. You can add chemicals and you can destroy it. Okay, you can get the meat to be tenderized by adding a chemical to it, all right, which makes it easier to manipulate and cook with. To that end, we have 4 basic levels of structure when we look at a protein. All right, you could see them listed right there: primary, secondary, tertiary, quaternary. 1, 2, 3, 4. That's what those words mean. With each level of complexity, we get more emergent properties. So we get a much more specific type of function out of a secondary protein than out of a primary protein. Okay, and conversely, tertiary and quaternary as well. I'm going to underline a few things here. Okay, primary level of protein structure is just a linear sequence of amino acids. Okay, just right here. Here's that primary structure right here. Okay, just the linear sequence of amino acids. All right, where would this happen? This happens in every single cell. Okay, you'll see this primary structure of protein. Okay. This is how DNA gets read. Okay, this primary level is the basic cookbook to how to make structure. All right, DNA gets opened up, gets copied. We take this linear sequence of amino acids out into the cytoplasm, and we can make stuff. All right, so this primary level is the building block for complex things in the body. For secondary level, you get these things called helices. A helix, H-E-L-I-X, is singular. Helix is singular. Helices is plural. Okay. You get this thing called an alpha helix and a beta pleated sheet. All right. When these primary levels become really long, okay, as this primary structure continues to be added to it, okay, it becomes really long, and it could overtake the size of the cell pretty dang easily. So it will start to kind of fold on itself or twist on itself. It's these hydrogen bonds, okay? As you can kind of see, I mean, this thing is really starting to kind of get really close to one another here, right? So now you're going to have those bonds, those those hydrogen bonds or those van der Waal forces really start to interact with that other chain that's getting really close to it. And it's those hydrogen bonds that help to form, as you can see right there, the dot dots, okay? The dot dots. Those hydrogen bonds allow this different shape to kind of manifest. So you get that kind of beta pleated sheet there, that fold, or you kind of get it to twist on itself. And that's the alpha helix there. Those twistings and folds help to give it this complex structure of like keratin in your hair. And that's what you see right here. Okay. It's this fibrous protein, keratin, that allows your hair to be either straight or curly. And it's your DNA, your specific read on how that specific collagen protein gets made in you, which makes your hair either naturally straight or naturally curly. Yes, I can manipulate that, right? I just apply some heat in a curling iron, and now I can change and I can alter the shape of the collagen fiber, and I can get it to curl up. All right. As you can kind of see there, it's also kind of the reasoning in behind the complexity of that spider web as well. Oh man, my nose. You get a number of helices, you get a number of kind of these beta pleated sheets, and you start to get a very complex structure as these things start interacting with one another. Okay. And here you can see there's a number of helices, there's some beta pleated sheets and folds here. Okay. So now we get this three-dimensional shape because of the interactions between those alpha helices and beta pleated sheets. This complex folding allows us to start to have not only those hydrogen bonds still there, because they were just there with the second level, but here in this third level, I can start to have a number of various different interactions with one side of this molecule structure and the other side of this molecule structure, right? So this tertiary level, now I can start to have ionic bonds, and I can have covalent bonds between these different players here, okay? And you can see I can start to get a very kind of complex shape here. A lot of proteins in the body stop here. Most proteins stop here, okay? They get you that 3D shape, and this really gives you kind of the functionality of the vast majority of proteins, all right? Not every protein has to go to that fourth level of organization there, all right? And that's just a number of tertiary structures interacting to do a specific thing, okay? Hemoglobin is your example of that one, okay? Consists of more than one polypeptide. More than one tertiary level of organization. Okay? Because again, the vast majority of proteins stop here at that tertiary level. Okay? It's the shape. That's the function it's going to do because that's the shape it does. Okay? Not everybody goes to that fourth level. Only a handful of things really go to that fourth level of organization. But now, at that quaternary level, dude, it's super complex. Okay? Super complex. There's a video. Okay? I'll have you guys watch that. Kind of explains a little bit more of it just explains what we did here. Okay? It puts some animations in motion too for you. Why is this all important? Really, I wouldn't ask. This is slide 44. Okay, I probably wouldn't ask this on the exam. It just kind of shows you the importance of all this folding, all these various different levels. If a protein doesn't fold properly or do its little twist, doesn't form into its secondary to tertiary structure properly, it doesn't function. And sometimes, you can get various different, sickle cell anemia is just a misread in the DNA. Okay, there's been one specific amino acid that gets removed, totally changes the shape of the red blood cell. Okay, makes it sickle instead of circular. That doesn't kill you, but I mean that's not the best situation. I mean you're protected against malaria, but other than that, it can cause a lot of micro damage inside, can tear up organs inside your body here. Sometimes you get these things, these prions. These are just misfolded proteins. Okay, and this gives you this defect called human transmissible spongiform encephalopathies. Okay, mad cow disease, okay, is an example of TSEs. They're believed to be to have a role with Alzheimer's, I believe, with cystic fibrosis as well. So there's some misfolded proteins that can cause cystic fibrosis as well. So this concept of protein structure, which doesn't seem too important, it is important, okay? Because it can really it can change shape and structure, and it can cause some very debilitating things like mad cow disease. And then you eat the meat that the cow, and then you consume these prions, and now, you have it. So, and it's in a lot of animals within the within the animal kingdom. So it's pretty crazy. Our last biomolecule, guys, is nucleic acids. And we have two different types of here that we're going to be looking at. Being able to maybe explain the difference between these two, sometimes make a good short answer question on the exam. Maybe that's a hint. I don't know. DNA stores the genetic material. RNA reads the genetic material. And when we get into, mitosis, meiosis, transcription, translation in unit 3, I believe, unit 3. Yeah, unit 3. We'll dive into this a hell of a lot more, okay? I know you're excited. All right, so what makes up a nucleotide? Phosphate group, a 5-carbon sugar, that's what pentose is, remember, from way back at the beginning, and a nitrogen-containing base. These are the nitrogen-containing bases. Okay. DNA has its adenine, guanine, cytosine, and thymine, and RNA has its adenine, guanine, cytosine, and uracil. So there's the difference. Again, one thing different, right? Know them now because we're going to be talking about them ad nauseum later on in this semester. Okay, so remember these things now. It's the sequence of these little nucleotides, of these 5 different nucleotides that get you all these complex things that we have inside of us, okay? And that's kind of what it looks like. Here's what the nucleotides look like. I'm not going to have you, this kind of puts it all together here. Here's my pentose sugar, my nitrogen-containing base, which is just one of these. N is nitrogen, okay? So there's my nitrogen, there's my nitrogen, nitrogens, okay? And there's DNA, and there's RNA, 5-carbon, 6-carbon sugars. I think I have a bunch of words here, right, obviously, but I have a table here, okay? Being able to talk about the differences between DNA and RNA, again, always make a good short answer exam question. DNA, the sugar is the deoxyribose, okay? Deoxy, that should tell you that it's not there, okay? No oxygen. Ribose, okay? It has the oxygen attached. Bases, adenine, guanine, cytosine, and thymine for DNA. Replace uracil for thymine for RNA. It is double-stranded DNA, okay? Double-stranded, and it twists around on itself, so it does form that helical structure. Okay, there it is right there. There it is right there. RNA is single-stranded. Okay, there it was right there, just a single strand. All right. Adenine will always bind with thymine. Cytosine will always bind with guanine. Okay. If it's RNA, then that's going to be uracil. Okay. And I mean, this is all it is. Okay, that chart is everything in here. RNA single-stranded, DNA double-stranded. Okay, forms that little twist, that helix structure that we talked about. Okay, adenine always binds with thymine for DNA. Adenine always binds with uracil if it's RNA. The last thing that we have here, guys, is ATP, and we will see this a lot in the next unit. ATP's basic component parts are adenine, ribose, and the tri part. Here's your 3 phosphates. Okay. If I said ADP, how many phosphates do we have there? 2, right? That's di. If we have AMP, mono, 1. We don't usually get that far though. ATP and ADP, you're going to see quite a bit, especially in the next unit. All right. This is our high energy molecule. Okay. That's why it's the energy currency of the cell. All that energy. Where the hell is that energy at? I don't see energy here. Energy is in that little chemical bond there. Okay. So when I break that chemical bond, when I make ATP into ADP and an inorganic phosphate, I get all of that energy that was in that little squiggly line, gets released. And now cells can do work. Awesome. Okay. Add water, destroy it, just like we did at the very beginning. Okay. How we basically put that thing back on is a pretty complex system of various different chemical reactions, and we'll talk about that in the next unit. Okay. Got to leave some mystery for you to keep coming back, right? That's why it's the energy currency of the cell. Okay. That's its only function. Okay. Hydrolysis, water to break down, again, gets you that ADP and an inorganic phosphate, and that's how it's written. That's a P. It may not look like it, but that's a P and a little I, and that tells you inorganic phosphate. Okay. And what does energy do? Energy is going to allow the cell to do work, and it has to do a lot of work. Just to sit here and do nothing takes a hell of a lot of energy, okay, for those cells. Where do we get this ability to make all that energy? From our macros: carbs, fats, proteins. Okay. We're not really concerned with nucleic acids in terms of our diet providing us energy. The lab today that we have listed on the schedule will allow you to kind of put this, what we did today, into use, into real world use. So make sure you don't wait too long before you do that and you dump all this information out of your head. Is there, I don't know if there is a Lilith Michaela Jackson here within the group. Please stick around. I have a question for you. Two things before we go here, guys. The first thing, type your name into the chat box, please, so I can make attendance. You can get points for being here today. Okay. Second thing, guys, if you have any questions, feel free to stick around and ask. If not, and you've typed your name into the little box, have a fantastic day.